Hello everyone, welcome to another video of DNA of Bioscience. Today we are going to discuss about the comparative analysis between all the 4 hypersensitivity reactions. So let's look into the points. The first point is the type 1 hypersensitivity reaction is mediated by IgE immunoglobulin. Type 2 hypersensitivity reaction is mediated by IgG or IgM immunoglobulin, but particularly IgG immunoglobulin is mediated. Then the type 3 hypersensitivity reaction, it is mediated by the immune complex. Type 4 mediated reaction is mediated by cell. Next point is the nature of antigen. What is the nature of antigen which is present in the type 1, type 2, type 3 and type 4? In type 1, the nature of antigen, it is soluble and exogenous. In type 2, it is a kind of surface antigen and it is also called the cell-associated antigens. In type 3, it is also soluble antigen. In type 4, it is soluble and cell-associated antigens. The next point is the immune reagent. Immune reagent means the reagent which causes the reaction. So in type 1, IgE immunoglobulin causes the reaction. In type 2, IgG. In type 3 also IgG. And in type 4, as we have discussed, it is cell-mediated process. So the cells, particularly the T helper cells and the cytotoxic cells involved in the reaction. Then the fourth point is the time required for the manifestation. In type 1, as we know all, it is also called immediate hypersensitivity. So it takes minimum minutes or hours. Type 2 hypersensitivity reaction, the time is totally variable. In type 3, it takes 1 to 3 weeks after exposure. In type 4, it takes 2 to 7 days after exposure. Then the mechanism, as we have already discussed, the IgG anti-IgG immunoglobulin, when attached with the allergen, they attached with the mast cell surface and they cause the degranulation of the mast cell and release the histamine, which result in the cell lysis. In the type 2 hypersensitivity reaction, the opsonization occur and the phagocytosis of cells occur. In the second step, that is in the ADCC mechanism, the neutrophil release a Fc receptor which attached to the Fc portion of the antibody. Then they release the opsonine and toxic products which degrade the cell. Then in type 3 reaction, the inflammatory cells complement activation, neutrophil enzyme degrade the cell. The key point to remember is FDI. First, the formation of the immune complex, then the deposition of the immune complex in the wall of the endothelial cell, then the inflammation which release the cytokine and activate the complements, and the enzymes then degrade the cell, particularly the lysosomal enzymes from the neutrophil, they degrade the cell and cause the cell lysis. In the 4 type of hypersensitivity reaction, the macrophage engulfs the antigen and they represent the antigen in the surface by MSC class 2 proteins. Then the T helper cell with the CD4 sense the antigen and they bind with it and secrete the cytokines which result in inflammation and cell lysis. But in the T cell mediated reaction, it causes direct cell killing. The examples of type 1 hypersensitivity reaction is anaphylactic shock, allergy reactions, asthma, atopy. In type 2, Rh incompatibility, that is the erythroblastosis fetalis. In type 3, rheumatoid arthritis, systemic lupus erythematosus. In type 4, the dermatitis, diabetes, and also in case of transplant reaction, suppose some tissue transplant or bone transplant. And the next step is the antibody is involved in type 1, type 2 and type 3, all the 3 hypersensitivity reactions. But in type 4 hypersensitivity reaction, no antibodies are involved, only the cells are involved. Then the mediators of the reaction are, in type 1, the eosinophil mediate the reaction. In type 2, the antibody which is involved, that is IgG, and the complement mediate the reaction. In type 3, complement and the neutrophil are the mediators, and in type 4, the lymphocytes are the mediators. Then the next point is name. What all name is given to the hypersensitivity reaction? Type 1 hypersensitivity reaction is also called as immediate hypersensitivity reaction, as it takes very less time to develop the response. In type 2 hypersensitivity reaction, it is called antibody mediator or called cytotoxic hypersensitivity reaction. In type 3, it is called immune complex mediator hypersensitivity reaction. And in type 4, it is called delay type hypersensitivity reaction because it takes 2 to 7 days to occur after the exposure with the antigen. Then the key words optional. In type 1, you can just remember the immunoglobulin is IgE. Mast cells are involved and they cause the degranulation which release the histamine and cause the cell lysis. In type 2, IgG and IgM involved. These are surface antigens, opsonization, phagocytosis occur, and ADCC mechanism also occur. In the next step, the immune complex is formed, then it deposited in the wall of the endothelial cell, that is the diffusion, then inflammation caused the neutrophil enzymes to released and they result in cell lysis. In type 4, mainly the cells are involved, particularly Th1 helper cell, CD4+, CD8+, CTL, antigen presenting cell, macrophage, and the example is poison ivy, which contains uracil chemical in the leaf, which is behave like a hapten. So that's all about the comparative analysis of the 4 hypersensitivity reaction. You can remember by simply remembering the type 1 immunoglobulin is involved, that is IgE. In type 2, 2 immunoglobulins are involved, IgG or IgM. In type 3, as I have discussed, FDI you can remember for 3. Type 3 hypersensitivity reaction, 3 letters FDI. And type 4 you can easily remember as it is the last type of hypersensitivity reaction. So that's all for now. Thank you.